Okay, so hello everyone. Today I am your moderator, and here we are going to pay attention to the presentation by Andini Fajarwati about. Wait a second. Plant tissue culture. For Andini, please.
All right, thank you, Fariza. So I will share screen. Okay, so does my screen appear?
Yes.
Okay, so today I like to talking about what plant tissue culture. So we begin for the definition of plant tissue culture. What is plant tissue culture? The plant tissue culture is defined as culturing plant seeds, organs, explants, tissues, cells, or protoplasts in synthetic genetic material that are chemically defined under sterile as well controlled conditions such as light, temperature, and humidity. Plant tissue culture, also known as micropropagation, because it involves rapid multiplication of small amount of plant material to produce more and more offsprings. In micropropagation, it refers to in vitro multiplication or plant reproduction under aseptic and natural conditions controlled to produce thousands or millions of plants to be transferred to the field. So as we know that plant has a totipotency properties, which is the ability of single cell to divide and produce all the differentiated cells in an organism. So plant tissue culture can be done easily. So there are six type of plant tissue culture. The first one is seed culture. So what is seed culture? The seed culture is the culture that using expand, which is seed, to be form a new individual. So expands is like the part of plants that we are used to undergo the micropropagation process. As well as embryo culture, it use embryo to form a new individual. And then for the callus culture, callus is unspecialized, unorganized, and the differentiated mass of cells arising from explants under in vitro condition. So in callus culture, it depends on the species that we like to do micropropagation. And then for the organ culture, it use organ as explant. The organ that use such as any part of plant like root, and then stem, and then leaf, and then the type one is protoplast culture. Protoplast culture is the explant is obtained from neck cells. So it the it the explant comes from neck cells, or as we know that protoplast is a cell that doesn't has cell wall. So it can do protoplast culture. And then the last one is anther culture. Anther culture is culturing anther or a part of the bud. So the unflowered flower, like the bud is used as explants to grow new individual. So the product example is Pomato and farming of orchid. Actually, there are more example, especially for the plants that extensively used in our daily life, such as we can farm the potato. We can also farm the banana and etc. So the next is the process, the micro propagation. Do the for 1, 2, 3, 4, 5 process. So the first process is stage 0 or pre-propagation. In this stage, the plant that we would like, the parent plant that we would like to be, that we would like to take the part of its plant will be selected because the purpose of plant tissue culture is to producing the better plant or the better individual. So it has to select the best of the best from the mother plant. So in this stage is selection step. The second stage is stage 1 or initiation of explant surface sterilization, establishment of mother explant. So in this process, when we already choose the best of the best plant that we would like to multiply, we choose the part of the plant which can be called as explants. So we choose the explant of the mother plants, and then we sterilize it in laboratory to undergo the next step. And then the stage 3 is subculture for multiplication or proliferation of explant. So in here the explant will be moved into the medium to growth root or other parts of the plant. So after stage 1, after it already sterilized, it will place in the medium, culture medium, and then the next stage is stage 3, which is shooting and rooting of the explant. So explant will be growth its root and other parts of the plants. And then after the plants that already growth, we call as plantlets. Plantlets will undergo the stage 4, which is hardening or weaning. In this stage, the plantlets will be placed in the ground in the new environment, not in the lab again. So it will move in the wide environment. So we can see it is just the plant that we already multiplied is can be adapt or not. If it cannot be adapt, it will die. and if it can be adapted, it will grow perfectly like that. So yeah, I think that's all for the I'm sorry. For the example, such as Pomato, this is the picture of the Pomato. So Pomato is potato and tomato. So we can also what fuse the two different germs of plants in protoplast culture. So it will produce the new individual from the mother, from the parent, such as Pomato or potato and tomato. Like that. So yeah, I think that's all from me. Maybe for the next session is question and answer.
Okay. So for everyone else, do you guys have a question? All right, one more person, I suppose. Okay, so let's begin from Rahma first, and then Aisha second, and then Miss Tulip third. Okay, so go ahead and ask question.
Okay, thank you, Pariza, and also Andini for the great presentation. I want to ask about the plant tissue culture, especially in advantages and disadvantages of plant tissue culture, comparing to another biotechnology process. For example, comparing to genetic engineering in plants, what is the weaknesses and benefit of the plant tissue culture? So this is my question.
Excuse me, can you repeat that again? Rahma?
Okay, I repeat my question. So my question is simply say, what is the advantage and disadvantages of plant tissue culture comparing to the another biotechnology process? Can you catch my question?
Yes, thank you very much. Okay, next is Aisha. Go ahead.
Okay, thank you, Fariza, and thank you for Andini for the presentation too. So my question is there any specific characteristic of the plant to the micropropagation, or we can do it to all plants? So is my question can be heard clearly?
There's some lines that are cut off, so can you repeat that again?
Okay. Is there any specific characteristic of the plant to do the micropropagation, or we can do it to all plants?
Oh, okay, thank you very much. Okay, then next is Mr.
Okay, thank you, Fariza. Okay, thank you, Miss Andini, for the great presentations related with the plant tissue cultures. I would like clarify about the term in here. In your mind map, you say in vitro multiplication or plant reproductions under aseptic and natural conditions. May you explain more what is the aseptic and natural condition to grow plant tissue culture? Thank you.
Okay, thank you very much, Miss, for the question. Before that, thank you for everyone who has asked the question. Now, Andini, are you ready to answer the question?
Maybe you can give me some time to answer.
Okay, then we will wait for the answer from Andini.
Maybe I can answer the question of Aisyah first.
Okay, go ahead, Andini.
Okay, so Aisyah's question is there any specific characteristic to do the micropropagation, or we can do it to all of the plants? Regarding to the stage, there is stage 0, which is the screening of the plant. So we choose best of the plant. If you ask, or we can do it to all of the plant, probably it can be done for all of the plant, but it has to undergo the step of screening. So we have to choose best of the plant, so we can produce the better plant, the better individual. I think that's my answer for Aisyah question.
Okay. Okay, thank you, Andini.
Okay. So for the Rahma's question, I think I will answer Rahma's question. Is the advantages and disadvantages of the plant tissue culture comparing to the other biotechnology method is, I think, plant tissue culture can be done easily because as what Ilva said, the plant has totipotency properties, which is the ability to of the single cell to grow into the new individual or in a new organism. So that's one of the advantages of the plant tissue culture comparing to other biotechnology method. And also the plant tissue culture, the new plantlets can be grown in a short amount of time, and only a small amount of initial plant tissue is required. So the next advantages is the new plantlets and plants are more likely to be free of viruses and diseases because it's done in laboratory. So it will be more sterile, and also we undergo the sterilization first. So it will be less of free of viruses and diseases. Also in advantages of plant tissue culture, there is no depend on the seasons, so we can do it all the time like that. So for the disadvantages of tissue culture, it costs a lot of money because it needs laboratory that can be a place to do this micropropagation or plant tissue culture, and there will be a failure in the plant. So in the stage 4 is like the plant is moved into the new environment after we grow it into the we grow it in laboratory. So the plant will undergo the seleksi alam, like that, the selection of the environment. So whether it can be growth or it can be died. So and then we know that before being cultured, the material is screened. So in this process is so crucial because we might do some mistakes, so we can pick any like abnormalities. So abnormalities could lead to the new plants being infected. So I think that's for the advantages and disadvantages of plant tissue culture.
Okay, thank you, Andini. That's all answer my question.
Okay. So the next question is from Miss Lilith. In vitro multiplication is plant reproduction and under aseptic and natural conditions controlled to produce thousands or millions of plants to be transferred to the field. So aseptic in here is the plant undergo the asexual reproduction that can produce the new individual or the new organism. And natural conditions is the natural conditions is we can control the natural conditions such as the light and then the temperature and then the humidity. I think that's what that I get from the journal, from my reference. Maybe if the other friends can help to answer, I really appreciate that.
Okay, thank you, Andini.
Okay, thank you very much, Andini. So now I think we are going to open the second Q&A session, and I invite everyone to go ahead and ask a question. If you have any question regarding the process of micropropagation, go ahead and ask. Okay, Hesti, go ahead and ask the question.
Thank you, Fariza, and also thank you, Andini, for your great presentation. So actually, I curious about the embryo culture. Embryo culture is only can do in the plant that has seed, and and I mean, can you explain more about the embryo culture and the differences between embryo and seed culture, Andini? I think that's all.
Okay.
Okay, thank you very much.
Can I answer it?
Excuse me, Andini?
Can I answer it?
Oh, go ahead.
Okay, so Hesti's question is the difference of embryo and seed culture. Yeah, embryo is like inside of the seed, so it will requires the plant that has seeds, and then for the seed is the explant of seed culture is comes from plants that are already cultured and grown under in vitro conditions. So if it's seed culture, it's seed itself, but embryo is the inside of the seed for the explant. Yeah, that's the difference of embryo and seed culture.
All right, thank you very much, Andini, for answering my question.
Thank you very much for the question. I invite one more participant to ask a question. So go ahead. Okay, Dea, go ahead and ask your question.
All right, thank you, Fariza. So I'm just curious about something, and I see questions. Andini answered that the plant mass has the best of the the plant must have the best of the best characteristics. So I just curious what means by best of the best characteristics. So what is the specific about the characteristic? Andini, thank you.
Okay, thank you for the
Okay.
question.
Okay.
Andini, do you want to answer it?
Yes.
Okay, go ahead.
So what I mean of best of the best is like the plants must be what bugar like that, like not layu, and then the quality of the plant itself, like the genotype, and then the what the the quality of the the what the buah so the like it should be has the good condition of leaf, and then the good condition of the part of the plant that we would like to undergo the micropropagation, or we can say that we would like to take as the explants must be in a good condition, like that, not like ada bolong-bolong ulat kayak gitu. I think that's all. I'm sorry.
All right, so it is compared by the each species compared to other, like it's compared in the between the specific species, right? Like ending.
I'm sorry, I cannot hear your voice there.
Also, it's compare between each species, like example, mango with mango, right? Which one has the best condition?
Yes, yes, that's right.
All right, thank you, Andini.
Yeah, you're welcome.
Okay, so I think that would be the presentation for today. So in conclusion, micropropagation is a process of which involves rapid multiplication of small amount of plant materials to produce more and more offspring. Here it is implied to be in vitro multiplication, or plant reproduction under aseptic and natural condition control to produce thousands of millions of plants to be transferred into the field. As you can see, there are several types of it, which is seed culture, embryo culture, college culture, organ culture, protoplasm culture, and anther culture. And then you have the product example being Pomato and farming of orchid. And the stages being stage 0, which is pre-propagation, and then stage 1, initiation of explants, and then the stage 2, subculture for multiplication, stage 3, shooting and rooting for the plants, and then stage 4 is weaning or hardening. All right, I think that's all from Andini. Thank you very much, Andini. That was a very wonderful presentation. I hope everyone can understand the points that Andini has to say. So thank you very much for listening. I think we're going to close this meeting. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.